Dr. Megan Hood graduated from North Carolina State University in 2019, and since that time she has developed a specialty in biosecurity and board health. She lives in New Ulm, Minnesota with her husband, where they have slowly converted to a colder climate. So, thank you.
Thank you. Clicker. Oh, thank you very much. All right, well, I will just jump right into it. Try to stand not in the way of the projector. So clearly, the spread of PRRS virus in feeds remains a hot topic. And over the past several years, mitigant use has expanded across the industry. However, out of the research that has been done regarding the spread of PRRS virus in feed, there has been some mixed results, which is what I want to talk with you all about today. I'll start with a couple of research highlights just to get into it, and then I'll move on to a project that I performed with Dr. Reicks and Dr. Kitt in 2021. So my first research highlight is a case study that I'm sure many of you in the room are familiar with because it was just released this last year. This was a study released by Drs. D, Clement, and Nelson, and described the infection of a 2,500 sow herd with PRRS virus following at a feed outage in October. So we're almost at October now. So come thinking to yourself about what those weather conditions likely were in October during this time, okay? These pigs exhibited PRRS symptoms 5 days after that bin was refilled, and then 5 days after that, or 10 days after the bin was refilled, this feed was tested and was found to be positive for PRRS virus with CT values ranging from 26 to 29. Then these feed samples were given to pigs and they developed PRRS virus. My next research highlight is a study performed by Dr. Blom and her associates at KSU. This was a study in which they were looking at the survival time of PRRS virus in various feed matrices. And these were feed samples that were inoculated with PRRS virus at a level of 10 to the 5.4 TCID50. So this term, TCID50, has been thrown around a couple of times today. So I just want to make sure that everybody understands what that means. TCID50 refers to the median tissue culture infectious dose. And some of you in the room might be more familiar with CT values. So I put some of those relative comparisons here on this slide. So at 10 to the 5.4 TCID50, we're looking at CT values ranging from 12 to 13, okay? So in this study, they took these feed samples, exposed them to various temperature conditions over time, and then tested them again, and those positive samples were then tested further in order to determine the surviving virus concentration. So I want to draw your attention here, and I guess this clicker doesn't work. There it goes. It's very small. But these were the results for whole feed. And as you can see, at 37 degrees Celsius, the virus did not survive particularly well. At 23 degrees Celsius, we saw survivability out to 24 hours with a TCID50 of 1. And at 4 degrees Celsius, we had one sample that survived out to 72 hours with a TCID50 of 0.42. Now, the reason why I've shared these two research highlights with you all today is from my biosecurity brain. So I work with a lot of boar studs, and on those sites, we require that when that feed arrives, it has downtime before those animals are going to consume that feed, right? So with my biosecurity brain, I'm looking at these results from Dr. Blom, and I'm feeling pretty good about my biosecurity protocol. However, when I look back at that case study, and I see that I have virus that's survived out to 10 days, oh, well, now I don't feel as good about that protocol. So Dr. Scherson just now did an amazing job of describing his work as well as the work of others in this area. And as you can see from his presentation, we have done a lot of work in this area, particularly with other viruses, such as PED virus, Delta coronavirus, and African swine fever virus. And we know quite a bit about these viruses, even though we have a lot more to learn. And this is certainly the case for PRRS virus. It's not as simple as it would seem to be able to spread PRRS virus through feed. which is a good segue to my project that I'll share with you all today. So this was a project asked of us by a set of producers that were interested in putting a mitigant into their diet. And they wanted to know which one is going to be the most effective for PRRS and the most cost-effective for my business, which is a question that I'm sure a lot of producers have today. And so this was a study that included 9 groups of 4 individually housed pigs. We had 1 negative control and 2 positive controls. That included a PRRS positive feed control, in which the PRRS was mixed into the feed, and then a positive oral control, in which that animal was given a direct oral dose, which I'll describe in more detail in just a bit. And then we had 6 treatment groups, each of which received various mitigants. I'm not going to get into today what exact mitigants we used in the study, but just know that it was a variety of mitigants commonly used in the industry today. Each group contained 4 pigs, which came from 3 litters, 1 pig from each litter, and a 4th random pig. Each group did not contain any runts at all, and the pigs were approximately 6 weeks of age. This is what those animals looked like, and we wanted to make sure we didn't have any nose-to-nose contact in the barn. So we did have to do a little bit of construction, as you can see here, so that this was possible. The blood that we collected to use as inoculum was collected from a barn positive for 144 PRRS, and it was what we would consider a pretty hot infection, with CT values ranging from 16 to 19, with a TCID50 value of 10 to the 4th. However, if you were listening to Dr. Shershen's talk, you might be already thinking to yourself that, oh, we might not have enough virus here. But spoilers. So anyway, this blood was stored in the freezer until we were ready to use it. The feed was mixed in the following manner for those mitigants. Mitigant was weighed at a gram scale at the feed mill, and then feed was weighed in 5-gallon buckets using a fish scale on the site, which was really fun. And then the mitigant was mixed using a paint mixer. And then all of that feed was stored in separate trash cans across the barn. This is what our timeline looked like. So at day -7, we started all of our animals on their mitigant diets. Then on day -1, we tested all of them for PRRS PCR and ELISA. They came back negative. And very important, we withheld feed that evening because we were trying to mimic an emergency feed outage. And also, when we gave the infective dose to these animals, we wanted them to be hungry. We didn't want this food sitting around. We administered the dose on day 0 and then tested each consecutive day out to day 4. Now, an important note is initially we were only going to test out to 3 days. And so at the end of that 3rd day, the staff commingled the pigs by treatment group just to make it easier for them to feed the animals. But then we decided we want another day of results. So that's why that happened. So on day 0, I took a half a cup of feed into a disposable cup, and then I mixed 1 ml of that inoculum, or in the case of the negative control, 1 ml of sterile saline, into that feed. Again, with a concentration of 10 to the 4th, TCID50. If you want to know how I mixed it, it was pretty simple. I have my disposable syringe that I've used to pull up the blood. I drop it onto the feed cup and gently swirl that serum into the feed. And finally, we have our positive oral control group. And so this was very similar to how essentially you would pill a cat. So I had someone restraining the pig. I took my syringe of my inoculum as far back to the throat as I could, administered the inoculum, and then massaged the throat of the animal until it swallowed. All of the feed cups were then placed in plastic bags and put into the refrigerator because we wanted to simulate an emergency feed outage. And the feed mill that we were talking to told us that the minimum time it would take for feed to go from the mill to a farm would be 1 hour. And we put it in the refrigerator to try to simulate those colder weather conditions. So I wanted to talk a little bit about kind of the biosecurity workflow through the barn, how we had these animals spread out across the barn. This was a 2-room system with a very small farrowing room and a larger barn here. We put our negative controls right here so that they would be the first animals we could easily work with each day. Then our positive feed control put in this separate room. And the animals we viewed to be the highest risk, the positive oral control group, we put directly under this fan bank so that all of the air could be pulled outside of the barn instead of flowing over the rest of the pigs in the barn. So, see if my video works. All right. I start out my day getting all my supplies here, work with my negative controls. Now I'm disinfecting as I go, as I move between my different groups. And as I work through those treatment groups, before I handle those positive controls, I'm going to put on my disposable coveralls and booties over here in the corner. I'm going to work with my positive feed control animals. And then finally work with my highest risk animals, my positive oral control animals, and then exit to the shower. So here are our results. So again, day 0, all of our pigs were negative for both PRRS, PCR, and ELISA. And then as we move through to day 4, we see more animals in the positive oral control group that are positive for PRRS. And these are the only animals in this study that became positive for PRRS. Now, I do get this question quite a bit. Why only 3 out of the 4 pigs positive? Well, I will be the first to admit that if you have ever pilled a cat before, it is not always successful. And I did have one animal at the time that appeared to have spat out a small amount of inoculum, and it would seem that it was enough for that animal not to get infected. So again, I want to reiterate, no pigs that had PRRS virus mixed into their feed became sick, whether they had a mitigant in their diet or not. The only animals that became sick with PRRS were those that received a direct oral dose. So the first discussion point from this study is, of course, viral level, because we've seen now from other studies, since we did that initial one, in 2021, that they're using higher viral levels in this feed, such as what I mentioned before, 10 to the 5.4 TCID50, which when we're looking at CT values, would be CT values ranging from 12 to 13. And it is certainly possible to find samples with that virus concentration. However, I would say that it is more uncommon. So my question that I bring to you is, what is the probability that this high amount of virus is going to end up in our feed? The next discussion point is about mixing, because I mixed that inoculum into the feed. Is it possible that by mixing, I therefore increased desiccation and led to the death of the virus? Well, for this study, we wanted to make it as realistic as possible. And so we know that as feed is moved through our system from the mill to the barn, it's going to be mixed at every step of the way. So that's why we did that. However, I'm sure many of you are familiar with the ice block model. So perhaps we're concerned with ice being in our feed and somehow preventing that mixing process or somewhat insulating that virus from desiccation. Or perhaps not just in ice, but wet areas, therefore, associated with that ice. And so my first question to you is, how is that ice getting into our feed? Are we perhaps concerned about a contaminated feed truck with built-up ice on the back that is dropped into our mill and somehow remains in that form throughout that process and makes it to our farm? And if so, what is therefore the probability that that high level of virus is in that ice? And finally, because I'm from North Carolina, for this model, I always like to consider climate, because compared to the Midwest, North Carolina does not nearly get the amount of ice and snow that we would see here in Minnesota. So along that line of thinking, are we then saying that our friends in North Carolina are off the hook? Except, remember from that case study that I pointed out from the very beginning of the study that happened in October during relatively mild temperatures. So, therefore, I have questions about this process, and I hope you do too, because there are a lot of questions that we need to answer about the spread of PRRS virus through feed before we can really feel confident about how this is happening.
Thank you very much.